In this section of Chapter 5, we look at membrane structure and function. We'll review a little bit about diffusion, osmosis, and then we'll look at how membranes allow cells to selectively bring in certain substances, but not everything. We covered this briefly when we discussed membranes being semi-permeable. In this image, if you recall, we were discussing the variety of functions proteins may have. Here, for instance, we can see that enzymes are facilitating reactions. An initial reactant comes in, a product is produced, that product is used by another enzyme, which then creates another product itself. Ultimately, triangle was being turned into this green thing, looks like maybe a rectangle. But in order to do so, it had to go through this step of being turned into a circle first. So we will at some point discuss the function of enzymes. They are pretty amazing. The function that I want to talk about in this particular section of Chapter 5 would be the function of transport. So here you're looking at one transport protein that's allowing a substance to go through. And here we're looking at another transport protein, and it is also allowing substances to go through, but notice that this one seems to be requiring a molecule called ATP. ATP is energy, and in the second section of this chapter, we'll explore energy concepts that address ATP. So brief summary of diffusion is that diffusion is basically a spreading out phenomenon, right? Diffusion is a large concentration of particles moving towards an area of lower concentration until reaching equilibrium. What we'll learn in this section is that this diffusion is also an example of passive transport. So what is passive transport? It's the transport of substances from high concentration to low. If it's happening through a membrane, we call that passive transport because no energy is required. It is passive. So why is diffusion across a membrane called passive transport? Again, it's because substances are just simply moving from high concentration to low. No energy is required in this case, and also no transport protein is required. Here's an example of what I mean. So let's say that we have a membrane here, okay? And we also have a variety of substances collected on one side of the membrane, very few on the other side. If those substances were simply to travel through the membrane from one side to the other, from high concentration to low concentration, that is a passive style of transport. And those substances went through the membrane without any help whatsoever. There was no protein involved, no protein transporter required. Things simply moved across. So what sorts of things can move across a membrane without any help? Well, things like oxygen, something that our cells need, right? We breathe oxygen to provide it to our cells. We're going to learn later that that's for the purposes of something called cellular respiration. We also generate things like CO2. CO2, the gas produced as a product of respiration, that can move outside of our cells. And a third item that can go through the membrane without any assistance whatsoever is water. In fact, water can go back and forth depending on the situation. Sometimes we have more water than we need, other times there's not enough water. Regardless of the case, water is constantly sloshing back and forth through membranes depending on the situation. But generally speaking, oxygen is usually coming in from high concentration to low concentration, whereas CO2 is moving from high concentration to low concentration, but notice again that they are moving in opposite directions of each other. Okay, so again, passive transport, is simply things moving from high concentration to low, and no assistance is required. There's no transport protein. In later portions of this discussion, we will see that some substances do need transport proteins, and they don't always move from high concentration to low. Sometimes they move from low concentration to high because our cells like to concentrate certain things. Okay, so here's a brief review of diffusion. So you can see a variety of substances here, right, concentrated on the left-hand side. This would be our semi-permeable membrane. It's only allowing certain things to go through, things that have the size to be able to get through this. If you were bigger, you would not be able to get through, hence the term semi-permeable. Eventually, things have moved over and back, by the way, right? They slosh back and forth. But overall, the natural tendency is for things to move from the left-hand side to the right-hand side until you reach, again, what we call equilibrium. There's an equal distribution of the substances on both sides of the membrane, even though things are still kind of sloshing back and forth. Remember also that different particles will diffuse independently of each other. These gold particles seem to be diffusing based on just their own concentration, whereas the purple particles seem to be diffusing based on their own concentrations. And in the end, both particles will have reached equilibrium.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Great. So I think that sort of seals the deal there, right? Things are just moving from high concentration to low, and you can see that there is no transport protein anywhere in here that oxygen and CO2 in this model require to be able to get through. They just simply go through the membrane. So why is it that they can get through the membrane? Well, oxygen is not polar. CO2, not polar. Remember, what that means is that they do not carry a charge. And this is important because if they don't have a charge, then that means that they can get through this portion of the membrane, which is non-polar. Because it's non-polar, it doesn't mix with things that have a charge. Oxygen and CO2, fortunately for us, don't have a charge, and that's why they can get through the membrane without any assistance.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Okay, so that's pretty important. Let's make a distinction here. What sorts of things can get through the membrane? Things that do not have a charge. What sorts of things will not get through the membrane? Things with a charge, or things that are too large. So we'll just make a quick note here that if you're charged or large, you cannot go through the membrane. But if you are small or have no charge, you go right through the membrane, no problem. So these small or non-polar particles are the ones that are participating in what we call passive transport. Sometimes I just simply like to call it passive diffusion to remind students that no energy was required. Okay, so the diffusion of water across a selectively permeable membrane, if you recall, is called osmosis. We defined this in lecture, and this would have been the lab exercises that we would have done during this shutdown. So if a membrane is permeable to water, but not to a solute, then only water can get through, and what this will eventually result in is in an equilibrium of water being produced on both sides of the membrane, even though the total volume of fluids may change. So let's see what this would look like maybe in a U-tube. So this is the U-tube we covered in lecture. So this is still sort of a review that we're going through. So notice again that on maybe side A, there is a relatively small concentration of particles. On side B, there's a much larger concentration of particles. Now the particles are too big to get through this semi-permeable membrane. However, water is small enough that it can get through. So in what direction will water go? Well, remember that if you have a small concentration of particles, say maybe you have 10% sucrose, table sugar, this really means that solution A has 90% water, right? If there's 10% sucrose dissolved in water, then 90% must be water. However, on side B, let's say that we have 20% sucrose. What this really means is that you'd have 80% water. And so now notice that there is more water on side A, less water on side B. Osmosis dictates that water would go from high concentration to low until reaching equilibrium. And here is the result of that equilibrium. The amount of water, total volume, has changed, but the concentration of the solutions on both sides of the membrane are now equal. So yes, the volume of water here declined, the volume of water here increased. However, on both sides of the membrane, you would have an equilibrium. You'd have 15% sucrose on this side, which means 85% water. And you'd have 15% sucrose on this side and 85% water. All right, so if anything, we can agree that the goal is maybe to achieve equilibrium, not to maintain volume. Like, we don't care about that.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
Great. So again, water simply going through a membrane is called osmosis, but osmosis, you've also learned, is an example of passive transport. Oops, passive transport. Sorry about that. Okay, so at this point, you should be able to write comfortably a short paragraph about what is passive transport and how does that relate to diffusion and osmosis. Okay, so here's some follow-up terminology that comes up when discussing osmosis. These terms only come up when discussing osmosis, okay? So remember that in our previous YouTube, we had a side A versus a side B. So I can replicate that scenario here in this form. Let's say that I had something like, I don't know, 30% salt on one side of the membrane, and I had, let's say, 20% salt on the other side of the membrane. Where would water go in this case? Well, if you have 30% salt on side A, this really means that you have 70% water. And if you have 20% salt in side B, you really have 80% water. All right, osmosis says that water goes from high concentration to low, The higher concentration is on side B, the lower concentration is in side A. So water goes from side B to side A. And as water is doing this, the concentration of salt in side A is being diluted, and the concentration of salt in side B is being concentrated because it's losing its water. So which side gained the water? Side A. Because side A gains water, side A is also gaining water pressure. So just think about a water balloon inflating as more water is being added to it. On the other hand, side B is losing water, and so therefore it's losing water pressure. So how do we describe the gaining of water pressure? Well, we would say that gain of water pressure is defined as hypertonic, which means high water pressure. Side B is losing water, so it's losing water pressure. So we would say it is hypo, as in lower or below, and tonic, water pressure. So literally low water pressure. And on side A, high water pressure. Okay, what if you have solutions, let's say side A, side B, and let's say that both solutions have something like 15% dissolved substances, glucose, sucrose, salts, anything you would like to dissolve. Well, then that means that they both have 85% water in this particular case. So water goes to one side and water goes to the other side at equal rates, and so you're already at equilibrium. So this sort of solution, these two solutions would be described as being isotonic. All right, so let's look at our definitions. Hypertonic means high water pressure. Hypotonic means low water pressure. And isotonic solutions are solutions that are equal in water pressure to both sides. And the only way for that to happen is if they both have the same exact concentration of dissolved substances. All right, so let's now look at why it is that these types of solutions are important to understand. In other words, let's see what kind of effect they tend to have on living cells. Cells will shrink in a hypertonic solution. Cells will swell up or bloat in a hypotonic solution. So let's look at models of that. Okay. Let's focus on the animal cell, and let's compare that to the plant cell. Remember that plant cells have something animal cells don't have, and that is this really cool, sturdy cell wall. Animal cells don't have that, and for that reason, they respond differently to different solutions. So let's look at a hypotonic solution. What is a hypotonic solution? It's a solution that's losing water, right? So if it's losing water, so here's the solution. If it's losing water, it's losing it to the cell, right? The solution outside with its dissolved substances is losing water to the cell through the semi-permeable membrane of the cell. And as the cell gains water, the cell is described as being hypertonic, right? So remember in our previous slide, if one side of a membrane is hypotonic, the other side must be hypertonic. You cannot have two hypertonic solutions separated by a membrane. You cannot have two hypotonic solutions separated by a membrane. You can only have two isotonic solutions, right? So if one side is determined to be hypertonic, then the other side must be hypotonic. So here we are told that a solution is hypotonic. That must mean that the cell itself is hypertonic. Now let's look at an isotonic solution. An isotonic solution is, again, a solution that is equal on both sides of the membrane, right? Two solutions that are equal in dissolved substances. So in this case, the amount of water being lost by the cell is the same amount of water being gained by the cell, and so the cell would appear to be normal. Okay, but what happens if you're in a hypertonic solution? This would have been the cool part of lab. We would have taken blood cells and exposed them to a salty solution, and we would have seen that cells shrivel up. So a hypertonic solution is a solution that's gaining water, and it's gaining water because it's really salty. So the water leaves the cell, and the cell becomes shriveled. Okay, so what would we describe the cell as if it's losing water? We would describe it as being hypotonic. Remember, if you say one thing is hypertonic, like the solution out here is, then that must mean that the other side of the membrane will be hypotonic. You can't have one without the other. The only time that you can have equal pressure or equal dissolved substances on both sides is when you have an isotonic solution. Okay, let's now compare the same exact solutions, but this time to a plant cell. Remember, the plant cell has that cell wall, so this is going to change the way plant cells respond to these solutions. So first, what happens when a plant cell is placed in a hypotonic solution? In that case, that means water is coming into the plant cell. And whereas an animal cell would rupture open and die, we call that lysed, when you rupture open, when you tear open. A plant cell doesn't tear open, again, because of its cell wall. In fact, it's normal. We describe it as being turgid, which is sort of like saying bloated. But they like that, whereas we don't. In fact, we die. An isotonic solution, on the other hand, for a plant cell, means, again, water goes in at the same rate as it's leaving, and the cell appears to be a little bit flaccid, maybe looking like it's beginning to show signs of wilting. But it's not fully wilted, right? It's just kind of soft. And finally, in a hypertonic solution, a really salty solution, the cell membrane, I don't know if you'll be able to appreciate this subtlety, but the cell membrane is starting to peel away from the cell wall. And there's these air spaces here left behind. And the water is leaving the plant cell. The plant cell is responding by shriveling up within the cell wall. See, the cell wall stays put, but the cell membrane begins to peel away. And in this case, we say that the plant cell is being plasmolyzed. Its cell membrane is peeling away from the cell wall. It's shriveling, and that's no good. That's full-on wilting. That's a dehydrated plant, and it will look like it. It'll be droopy, flaccid, and giving you all kinds of indications that it needs water. So let's take a look at videos of these moments. Okay, so here's a plant cell. This is Elodea. This is the same plant that you looked at in the cell structure and function lab. And you can see here the cell wall, and you can see the cell membrane peeling away, leaving behind air spaces. And you can see it all over the place. You can see all these air spaces being produced by these dehydrated plant cells. Here's something also noteworthy. Notice that all the chloroplasts are being dragged to the center of the cell as it peels away, right, from the cell wall. So all of that cell membrane, as it's dragging away from the cell wall, it's bringing together all the chloroplasts somewhere in the middle of the cell. Again, this would have been really cool to see in lab. All right, let's look at the opposite effect. Let's look at what happens when a plant cell is nice and bloated. It's going to love it. It's going to be looking very healthy. All right, look at these chloroplasts. The chloroplasts are doing really well. You can see the cell wall here of the Elodea, and the cell membrane is like saran wrap just beneath the cell wall. And then you can see the chloroplasts are swimming around. They're loving this. There's all kinds of fluids for them. So they're doing pretty great. This is a really, really happy plant cell. And you can also see a bunch of little single-celled pond water creatures moving around also. Okay, so again, this plant cell would be described as being turgid or bloated, and they love it. We don't. When we're turgid or bloated, we have things called hypertension. All right, so let's get back now to the transport of substances through a membrane. So we're no longer talking about things like oxygen, CO2, or water, things that can move through membranes without any help, things that are part of what's called passive transport. This is now a separate type of transport. This is now something called facilitated diffusion or facilitated transport. If you speak Spanish, you probably recognize that term as suggesting fácil or easy. And that's exactly what we're trying to suggest here. Transport proteins make the transport of difficult substances much, much easier. All right, so do you remember the characteristic of substances that cannot get through a membrane? They are things that have a charge or things that are very large. All right, sometimes cells need these things, like we need sugar, and sugar happens to be large. And we also need ions like sodium and potassium and chlorine. And even though those are little teeny tiny ions or single atoms, they have a charge, and that makes it difficult to get through a membrane. So if you're hydrophobic, those can get through a membrane easily. But if you have a charge, or if you're very large, you're not going to be able to get through. In those cases, we need to employ something called facilitated diffusion. Again, because it's diffusion, it does not require energy, and it relies on substances moving from high concentration to low. Okay, so in facilitated diffusion, we're going to need transport proteins. One example will be an aquaporin. I'll show you a little of that in a second. So I'm going to draw a membrane here. Really bad. Wow. Okay, but let's say that I want to bring in something like chlorine. Well, chlorine has a negative charge, right? You wouldn't know that in advance. Chlorine is just one of many, many ions out there. So if I ever ask you if something can get through a membrane or not, and you find that it has a charge, like say calcium has a charge also, it has a 2+ charge. In those cases, because you see the charges, you would say, those things cannot get through the membrane on their own. They cannot get through the membrane through passive transport. Those things are going to need specific transport proteins. So there's a specific transport protein that allows chlorine to go through. There's a specific calcium transporter that allows calcium to go through. If you were really big, like glucose, and I'm not doing it justice here, it would be much, much bigger than any of these that I've drawn. That one would need its own transport protein to go through the membrane also. Okay, so they are still going from high concentration. This is my symbol for high concentration, high, and then the brackets concentration, high concentration. And they would still move towards low concentration. Right? But you need to have these transport proteins to help you accomplish that sort of movement. And did you notice that the chlorine could not use the calcium transporter, and the calcium could not use the chlorine transporter? The transporters are very specific as to what they allow to go through. So this one's only for glucose, this one's only for calcium, and this one's only for chlorine. But again, in every single case, everything is going from high concentration to low, and you need a transport protein. So that defines facilitated diffusion. I'm going to be reviewing this at the end of the presentation. So now let's take a look at an example of aquaporins. Aquaporins, as you might be able to read into, aqua means water, and porin means a pore. But in this case, the pore is being produced by a protein itself. So it's like a protein channel or a protein transporter. Okay, let's take a look at this diagram. So here is a transport protein in purple, And notice that it's allowing a specific solute to go through. The triangle cannot go through this. The triangle would need to have its own separate transport protein that allows it to go through, but that same one would not allow the circle to go through. So transport proteins, I want you to understand, are very, very specific as to what they allow to go through. And this way, cell membranes can determine and select what goes through and what doesn't. Again, cell membranes are described as being selectively permeable for this sort of reason. Okay, let's look now at a third type of transport. We've covered passive, facilitated, and now we're going to look at active. And as active transport is described, I want you to pay close attention to the term active. This means that energy is being used. And what is the energy molecule of the cell? It's ATP, and we're going to learn about that one in the second presentation for chapter 5. All right, so in active transport, a cell must expend energy to move a solute against the concentration gradient. This is why you need energy. You're not going from high concentration to low. You are doing the reverse. You're going from low concentration to high. That's like maybe sitting in a small boat that's taking in water, and you have to spend your own energy to take that little bit of water that's going into the boat and basically dump it back into the ocean or the lake, which has the higher concentration of water, right? So water just wants to go from high concentration to low. It's going from the lake into your boat, and you don't want it to. So you're going to take the water from your little boat, and you're going to scoop it out and throw it back into the lake. That's going from low concentration to high, and you have to expend energy to accomplish that. All right, so let's look at an example of active transport and then maybe a video.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so for those of you moving on to anatomy and physiology, the sodium-potassium pump is super important. Sodium-potassium pumps are essential in the understanding of how nerves operate and how nerves, in effect, will control muscle movement. You'll learn about action potentials and membrane potentials in that course. So you may want to pay extra attention to this particular sodium-potassium pump. So why was it called a pump? Well, because you were pumping things from low concentration to high. That's what all pumps do. They need energy because you are asking them to move things to a higher concentration. So if you noticed earlier, you had very few sodium ions here, and they attached themselves to the transporter, and then ATP came by, dropped off a phosphate, and then that changed the shape of the protein so that the sodium could move to the outside. So our cells like to concentrate salts outside the cell, and we like to concentrate potassiums inside of our cells. Okay? That is the sodium-potassium pump's job. But it cannot work unless you provide energy. So, what is the major difference between active transport and the other two transports, passive and facilitated? The major difference is you are moving things from low concentration to high. That's the major difference between active transport and the other two. There are some things that are similar, however. Did you notice that active transport requires a transport protein? And so does facilitated diffusion. They both need proteins. Passive transport doesn't need proteins. That's because, remember, oxygen just goes right through the membrane, CO2 goes right through the membrane, and then water goes back and forth, sloshing around. So passive transport, no protein. Facilitated diffusion, maybe transporting calcium. That requires a transport protein. So does active transport. But you should be able to write comfortably the similarities and differences between passive, facilitated, and active transport. Those are three types of transport mechanisms I'd like for you to know in time for our second unit exam. All right, and finally, another example of active transport would be exocytosis and endocytosis. This is the transport of things in bulk, right? So we have been seeing how things move across membranes individually, but sometimes cells want to just transport things in bulk, right? There are times when you can go to the grocery store and buy, one or two things, and then there's times when you go to Costco and you buy stuff in bulk, right? Say toilet paper. You could buy, I don't know, a couple of rolls or something like that at the grocery store, but if you go to Costco, you just buy everything. Apparently, is what's happening nowadays. You buy a big package that has, I don't know how many rolls in it, but that would be an example of bulk. Okay, so what is exo? It means exit. What is endo? It means enter. So to exit the cell means exit the cell. To come into the cell means endo the cell. All right, so let's look at some sort of diagram of this. Well, before that, let's look at specific examples of endocytosis. When a bacteria is swallowed up, so here's a little bacteria. It's got flagella and fimbriae. When a bacteria is swallowed up by a white blood cell, these are the cells that are part of our immune system. When it swallows a white blood cell, that's called phagocytosis. Phagocytosis is like cell eating, right? And now that bacteria is inside a food vacuole. This should sound familiar. We covered this briefly in class for Unit 1. Another example of endocytosis would be receptor-mediated endocytosis. So let's look at examples of that. All right. Here's phagocytosis. All right. Here is some food item. It could be a bacteria or it could be something else. And notice that the membrane of the cell folds outward, kind of like reaching out to engulf, to bring in this substance into the cell. Now here's the food vacuole. Massive bag loaded with food. Phagocytosis for food. Receptor-mediated endocytosis is bringing in large substances also, but it is very, very selective. In this case, notice that it's got these little red receptors, and those red receptors bind specifically to the purple triangles, not to the green circles, so that only those triangles can bind to the receptor. But when you bring them in, notice that some of these little green circles kind of piggyback for the ride, and they also end up coming in. All right, but the goal was to bring in the triangles, right? But by accident, the cell also brought in these guys, the little green circles. So what do you think about how selective a cell is when it uses endocytosis? It's not very selective, right? Because it wanted to bring in only the triangles, and it ended up bringing in also the green ones. And we never saw that in the other type of transport mechanisms we described. All right, so let's do a summary. All right, here's what you should know. You should know what diffusion is. Also, you should know the factors that affect diffusion. I apologize for how horrible I'm writing. I think I can get better. The factors that affect diffusion, if you remember from our lecture, was temperature, size, the medium, and the concentration gradient. So we didn't describe that in this particular lecture, but I did in a previous one when we last met in lecture. So I want you to go back and review those notes, or you can read about them in your textbook. All right, the other thing you need to know is osmosis. And you need to remember that osmosis is like diffusion, but it's specifically the diffusion of water. And if you should ever be given a side A versus side B scenario, like 8% salts here, 12% salts here, you should be able to tell me where water goes, right? Water goes to this side, and then you should be able to describe to me which side is hypertonic and which side is hypotonic. This side gained the water, this side gains water pressure. This side lost the water, this side loses water pressure. Right? So I need you to understand these terms, hypotonic and hypertonic. And the only time that you will see isotonic solutions is when both sides have the same amount of dissolved substances, like say 10% salt on that side and 10% salt on this side. All right? So I need you to know diffusion, osmosis, the factors that affect diffusion, and then I want you to know the terms of tonicity associated with osmosis and be able to determine what side water moves to if given two different compartments. And then we can focus on transport mechanisms. So let's do a quick outline of what we've just covered here. In transport mechanisms, we covered passive transport. These are things moving through the membrane from high concentration to low, right? So this is diffusion, osmosis. If you're moving through the membrane without any help whatsoever, we said oxygen can do this, CO2 can do this, and we said water can do this. And why can they do it? Because they are small and do not have a charge. Okay? So that's one type of transport. The other one is facilitated. So what sort of things need facilitated transport? Slash diffusion. Things that are tiny but have a charge, those are going to need help. Facilitated. I need help because I have a charge. Examples, we discussed chlorine, calcium. We saw examples of sodium, potassium, right? All of these things that have a charge, all of those are going to require transport proteins, even though they are tiny, right? Chlorine is tinier than water, but chlorine has a charge, and so it's got to go through. Some of you must be wondering, hey, doesn't water have a charge? Isn't it polar? But remember that that's a partial charge. It's not there all the time. So when water is going through the membrane, it doesn't have the charge. It loses it. When it goes across the membrane, it's on the other side, then the charge comes back. All right, back to facilitated. So we said if you're charged, you're going to need help. And we said if you are big, you're going to need help. Like if you are glucose, you're going to need a transport protein, right? So those guys need little tiny transport proteins or big transport proteins to be able to go through. All right, and finally, we covered active transport. And active transport includes the sodium potassium pump that we watched in the video, and it also includes endocytosis and exocytosis that we saw in some diagrams. And you want to remember that active transport is required because you are moving substances from low concentration to high concentration, or you need active transport if you are transporting things in bulk. Moving the membrane to that degree to swallow up substances, that's going to require energy. All right, so I hope you do well with that, and if not, if you have some questions, please remember to email, and then I can give you descriptions, or I can maybe point to certain diagrams in the textbook, or simply go back and review this thing, right? At that point, I'm just like that YouTube person who you can stop and rewind, whereas in lecture, it's hard to stop me and rewind me, right? So hopefully this works out for you. And I think I'm going to get better as I keep doing more of these. P.S. I hate my voice, so I'm not going to listen to this lecture again to see how I did. All right, y'all. Have a good one.